Alright, this it, man. This is our last selection now. Are we talking about the cancer? Now remember, cancer, it's just cells becoming more stem-like, right? They're going back to their original nature. That's really what a stem cell is. Some genes have changed. They've turned on or off, and now it is more like a stem cell. Genes have turned on or off, and now it is more like a stem cell. That's what cancer is. We have all these kind of ideas in our brain about what it is. It's just cells kind of not doing what they're supposed to be doing. They're like, I want to go back to my old ways. But no, you're supposed to be a skin cell. You're not supposed to be a stem cell. You're supposed to be doing skin cell stuff, for example. All right? All right. Let's talk about a couple terms here. All right. Well, if you get cancer, this is what you want. You want benign cancer. What is that? Well, it means that the cells are dividing. Cells are dividing abnormally, but that's it. Not a big deal. So you'll have a benign tumor, which means that the cells shouldn't be dividing in that way, but that's all they're doing. So it's not a big deal. They're not going to go anywhere. This is usually not life-threatening, depending on where it is. Like if it was in your brain, it's going to be life-threatening. So the cells divide, but they stay put. They stay put. Usually not life-threatening. Okay, and then we have malignant. This is the one you don't want to hear about. Malignant. Cells are abnormally dividing. And they've changed. Remember this? We go up here. They change identities and become different cells. So cancer cells multiply. If that's all they do, then it's benign. But now if the cells within there are also changing identities and becoming different cells, that's when you get a real mega problem. That's malignant. Cells are abnormally dividing and changing into invasive and mobile Crawley cells. Those are actually two different terms, Crawley cell. Invasive means kind of locally penetrating, but not pulling away from the tumor. Crawley cells means now I'm leaving the tumor behind. So invasive means invading nearby tissue, but staying attached to the tumor. Invading. Whereas Crawley means detaching from the tumor and going elsewhere. We'll talk more about those. Okay? That is called, well, let's talk about this. All right, cancer. Here's some cancer facts for you. 85% of cancers come from epithelial tissues. That makes a lot of sense. If you guys remember your epithelial tissues, we gotta go all the way back here to tissues. It was one of the 4 tissue types. It was epithelial. This means above or on top. Function is to line us, the outer layer, protection, absorption. So these are exposed to the outside environment. Your skin is exposed to UV radiation, poisons, toxins, chemicals. Your digestive tract is exposed to chemicals, alcohol, pesticides, so on and so forth. Red dye Lake 40 or whatever the fuck that is, right? So these are tissues that are exposed to what we call insults, things that can damage them. Look at that going all the way back there. All right, 85% of cancers come from epithelial tissues. So most cancers come from epithelial tissues. Okay, but they don't have to stay there. 90% of cancer deaths are the result of metastasis. Metastasis, or metastasis, if you want to break it down. 90% of cancer deaths, almost all of them, are the result of metastasis. And what is this? Metastasis is cancer cells crawling away. All right? It's this. They're crawling away and detaching from the tumor. All right, so this would be malignant tumor, benign tumor. All right, well, how do we get any of these kind of effects going on? Well, let me pull this up. Let's talk about a couple other terms, actually. So if we go back here and look at this, we only needed 4 genes to go from a fibroblast into a pluripotent stem cell. This is also like you only need a couple changes in genes expression, like genes turning on or off, to basically go from a normal cell to a cancer cell. That's essentially what that's saying. Like, you don't need a lot of big changes. Okay? So we got a couple of terms here I want to go over. We got something called oncogenes. Oncogenes. Onco means you go to the oncologist if you have cancer. This means cancer. And then we have tumor suppressor genes. Tumor suppressor genes. Okay, so oncogenes, if you get mutations in either one of these, they can result in cancer. So these oncogenes promote tumor formation when mutated, when turned on. So these are like what we'd call a gain of function is another word for these. Like you get this mutation and these now allow the cell to do something. Gain of function. I'm sure you guys have heard that, like with respect to COVID. You get this mutation, it turns on this gene, and now bam, you got cancer. Okay? Or like the early stages of what could then become cancer. Okay, tumor suppressor genes, these normally inhibit the cell cycle. They inhibit tumor formation. So if you get a mutation in these, the loss of function, that's what it's called, of the gene, leads to tumor formation. It's kind of confusing. I feel like I know I'm confusing you with that. But these things, you could think of these as they suppress. So they do things like normally inhibit the cell cycle if things are wrong. So then if we get a mutation in these, well, now they don't work properly. So even if something is wrong, they're not going to stop that cell from going through the cell cycle. And really, actually, these would actually be, let me move this over here. An oncogene would be, now we have that gain of function. A proto-oncogene is the thing that actually would be mutated. So this, if it is turned on when it shouldn't be, then it's going to become an oncogene. Okay? All right, so let's just kind of walk through this a little bit. Just to see if this makes sense. I'm going to pull this back up. This whole thing. This cell cycle, right? So we got our growth phase. Everything's good. Everything's good. We got a checkpoint here to make sure that the cell's ready to move on to the next phase. Okay? Like it's grown properly. It's the big enough size. Okay? We got checkpoint. Also then we copy our DNA. We grow. Copy our organelles, got a checkpoint, cool. Let's go into S phase. All right, now we make our DNA, make copies of our DNA, and then we go into G2 phase. And at the end of G2 phase, this is where the cell makes sure it's copied its DNA properly before it goes into M phase and divides. All right, so we would have tumor suppressor genes here that run this checkpoint, for example, to make sure that they've copied their DNA correctly. Well, let's say now we've got a mutation and this tumor suppressor gene doesn't work anymore. So now this cell can just freely go through the cell cycle even if it's not copied its DNA properly, which can then result in more mutations and more mutations. And now you've got, maybe let's say you've got 4 genes all messed up. And now you've got a full-on tumor. Maybe it's benign at first, but then it's accumulating more and more mutations, and maybe now it's even malignant. Okay? So know those 2 basic things. An oncogene basically, if you turn it on, it can allow the cell to go through the cell cycle more quickly. Or it can allow the cell to start to crawl. Or it can allow the cell to do some function better. Maybe it allows it to divide faster, or allows it to feed itself with blood vessels, and we'll talk about that. All right, so let me write down what I mean here. So if our cell cycle checkpoints get messed up, the cells can divide in an unregulated fashion without checking to see if the DNA has been properly copied. This can result in lots of cell division and additional mutations within the cancer. Does that make sense? I know you can't talk to me. I'm alone in my office on a Sunday. Okay, let's talk about a specific example of kind of how a cancer goes. Okay, so let's draw a little surface of our skin here. We got our epidermis. We've got our dermis. And we've got our subcutaneous layer, which is full of a bunch of fat and blood vessels. All right, so we got fat, we got blood vessels. Okay, and we've got our epidermis. We got our dermis, and we got our subcutaneous layer. Yeah, subcutaneous layer. All right. And let's say we got ourselves a beautiful little, it's actually a type of stem cell down here in our skin called our basal cells. And if you remember, our skin, this is where our basal stem cells. Basal stem cells. All right, and these guys divide, and these guys divide to make more skin cells all the time. So these guys are already like kind of primed to become cancer cells in a way, because they're stem-like. They continually divide to make this conveyor belt of cells. I don't remember if I talked about that. I think I did. Let's go back here to epithelial tissues. Did I talk about that? Nah, I didn't. That's okay. Talked about cell-cell junctions ad nauseum. That's cool. But if we look at, let me look at this. So these cells right down here would be your basal cells. These guys divide to make this cell, and then these cells keratinize, get harder with keratin, and then eventually they flake off and they become dust. And this is how your outside your skin stays or protects you. It's by continually dividing to make more and more of these cells. So these would be like the basal cells down here, which divide to make this, and then they kind of keratinize and become hard and dead up here. Okay? So now let's say, let's go back. Do some muscles. Whoa, 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 whoa. Yeah, look at the nuts on that boy. Okay, coming back. Okay. Okay, so now we're at our skin, and we got, bang, we out in the sun. We out in the sun, and boom, we're getting cooked in this Merced sun with some UV radiation, one of them insults, which is a carcinogen. This causes what's called thymine dimers, and then when the cell divides, if it's not properly fixed and trimmed out, this can result in mutations. Boom! And let's say we get one of these mutations, and now this basal stem cell done had a little change in it in one of its tumor suppressor genes, let's say. And now this guy uncontrollably starts to divide way too much. And it just keeps going, keeps going. So now we got unchecked cell division. There's no, we have lost a tumor suppressor gene, maybe also had an oncogene going in there. Division. Unchecked cell division. This caused a mutation. Unchecked cell division from mutation. Okay? Bam. What do we got here? What is this thing that I'm drawing? This is a tumor. And what kind of tumor is it? It's a benign tumor. Right? We just got this on, this cell just continues to divide. We got ourselves a benign tumor. Okay? And maybe through all of that unchecked division, what happens? let's see. Maybe now we get ourselves another mutation. Boom. Whoops. Boom. 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 Okay, and maybe it's even more rapid division. Even more rapid. Okay. Cool. All right. Now, let's say we get another mutation. Benign tumor, second mutation. Second mutation. Resulting in more division. Okay. Division. Now let's say we get a third mutation. All right, we'll draw this bad boy in this blue color, I guess. Okay, and this is no good. No good at all. Because now this cell is no longer staying. These are still all epithelial-like. They all stay with each other. Right? They're all connected. But now we got this mutation where this guy wants to crawl. So now we got a change in behavior mutation. And this has all resulted from this just uncontrolled cell division. Change in behavior mutation as a result of uncontrolled cell division. Something happened to my mitosis notes actually in this class. So I lost them. But if you think back to mitosis, meiosis, you know, what's going on with all of that, you know, if you just have unchecked cell division, DNA is not properly copied, chromosomes aren't properly separating, all this stuff can result, I have it right there, when there are problems with cell reproduction, this results in cancer. Or it can result in cancer. It doesn't always, but it can. Your body can come clean it up. All right? So this is happening. We've got these cells crawling around. And now let's say they crawl themselves over there, over to this blood vessel. Oh, where's this? Where do you want this to go? Where do you want that blood vessel to go? Well, let's see. Let's say it goes to this little guy. And let's say we go into a blood vessel near your brain. Right? And then let's say that this boy crawls out. And then let's say, then it makes a brand new home here. Boom. 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 And now we got a tumor forming in your brain. Okay. So now we have a secondary tumor in the brain. Okay, and then that's going to push on your brain and you're going to die. This pushes on your brain and you die. Fun, right? Very uplifting way to end the semester. Okay, this process of the cell breaking away, this is called metastasis. All right, metastasis. Once the tumor metastasizes and starts to crawl away, it can go anywhere in the body. You could be riddled with tumors. You could have secondary tumors then showing up in the brain, blood vessels. Once the cells get cancer cells, metastasized cells, metastasized cells get into the bloodstream, they can create secondary tumors anywhere in the body. They can get to your lungs. Boom. Now you got tumors in your lungs. They can get to your muscles. Boom. Now you got tumors showing up in your muscles. They can get to your digestive system. They can get into your lymphatic system. They can get into your brain. And this is really the cause of death from cancer. It's these secondary tumors. This will eventually result in death. Okay, and cancer is really kind of devious. Because when it's kind of in this second stage here, what it'll actually do, secondary mutation, let's say, and this is like what different stages of cancer are. They're each a little different, but like stage 1 versus stage 4. Like stage 1 would be like this, whereas like stage 2, 3 would be here, and stage 4 would be this is occurring. And every cancer is different. We're never going to find like a medicine that will cure cancer. It's not possible to do that because every cancer has different genes can be activated. There's, I don't even know how many different oncogenes and proto-oncogenes and different cell types that you're starting off with that can then have 1 or 2 or 3 or 4 different levels of expression of each tumor suppressor gene and different oncogene. All right, so let's say we're in this like kind of stage, middle stage here. We got the secondary mutation results in more cell division. And the tumor needs to eat. It needs to eat. It's going. It wants to eat. So what does it actually do? It'll turn on genes to bring in new blood vessels, right? Because it has this whole big mass of cells that's normally not there. This is called angio, meaning vessel, and genesis, meaning new, or to make. Angiogenesis. So it'll bring in the growth of capillaries to feed that tumor. I'm just drawing one little section of it. But all of this would have new blood vessels that grow towards it. So they'd be secreting hormones to grow in new blood vessels. So if you could stop this angiogenesis, you could kind of stop tumors in this like early mid stage. It's one kind of method for fighting cancers. Another popular one, I actually don't know how successful this is or how good we're at doing it, but a very popular way to fight cancer cells with chemotherapy is to affect this, especially anaphase. Because we have drugs that can inhibit microtubules. And so if you inhibit cell division, you can stop and thwart cancer. But you have cells that normally like to divide all the time. Like I said, our skin cells, especially our hair cells, are constantly growing from cell division. And our intestine cells. So when you get chemotherapy with something like Taxol that inhibits cell division by inhibiting microtubules, things like your hair fall out. And you feel very nauseated because your intestinal lining can't properly replenish itself. So it's a side effect of doing chemotherapy. All right, and this is the result of most of our death here. This is why metastasis results in 95% of cancer deaths. It's because now we got the cancer in our lungs, in our brain, wherever, in our mouth, our lymph nodes, everything. Okay, pretty cool stuff, pretty interesting. All right, I guess the thing that I want to talk on at the very end here that I want to hit on is it relates both stem cells to cancer cells. Pull up a little picture here of a lady. This is a picture of a woman named Henrietta Lacks, who had a type of, she had cervical cancer. It's Henrietta. Actually, I'll just pull up this other picture too. And we should all thank her because we've learned a lot about biology from her without her permission. So she had cervical cancer, and doctors took some of her cervical cancer cells and they grew them in a dish. And what they found is that Henrietta's cancer cells could grow forever in a dish. As long as we kept feeding them. They're still alive today. They're called HeLa cells, or Henrietta Lacks HeLa cells. And these are used in research. They're human cells, so in a weird way, she's still alive. That's why this book is titled The Immortal Life of Henrietta Lacks. Because her cells are immortal. They'll live forever as long as we continue to feed them, and we spread them all over the planet, and researchers use them. Normal human cells die after so many generations. Generations in a dish. They'll divide some and they'll be fine, and then eventually they die. Why? Why is that? Why can't you just grow any kind of human cell in a dish forever? Why? What is special about these cancer cells? What is special about these cancer cells and stem cells too that allows them to live forever? Live forever. It's a very interesting question. What is it about them? If you remember up here, one of the things that I talked about with cancer cells and stem cells is that stem cells remain youthful. Cancer cells remain youthful. That's what I mean by this. And this is actually a really fascinating thing that has to do with the way that we copy our DNA. It's a physics problem. Let me pull up this little video here. Well, if you remember me showing you this, but this is a DNA copy machine. We got one strand coming in here. One of those strands, as they're split, can be instantaneously copied. It's in the correct orientation. But DNA is actually reversed, so they're not going in the same direction. One is going in this direction, one's going in the other. So this one can be like copied easily. This one got to be flipped around, and that's what this one's doing. You see, it's got to flip. It's got to grab it. Watch it. Watch this right here. It's got to grab it and flip it around, and then it can copy it. But you see, there's these little gaps between it. It's basically in the wrong orientation to be able to be copied. It's not in the correct orientation. So you got to let it go out first. You got to get some slack, and then you got to bring it back, and then you got to copy it. All right? So we got ourselves a problem here when we get to the end of DNA. The very tippy end. And that's, we don't have any slack to be able to copy that other fragment. And so what happens if we look at our chromosomes, let's go to our chromosomes. All right, let's say we're looking at chromosome 1 here, like so, like that. At the very end, if we were to zoom in here, at the end, we got some specialized, just sequence of DNA. It doesn't really do anything other than act as like a little protective layer, like that. There's no genes in here. It's very similar to, if you look at your shoelace, let me take my shoe off here. you look at your shoelace, we got this little thing right there called an aglet. Okay? See that? It's just a little plastic wrapped around the fabric. And what that's doing is it's keeping this fabric from fraying out and getting ruined. It keeps it together. It's called an aglet. Well, your chromosomes have a little aglet as well. It's just made out of DNA. There's nothing special about it other than it's just a little protective layer. And every time, because of that physics problem I showed you, every time you copy your DNA, your little aglet gets a little bit shorter and 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 a little bit shorter. Let's say until you divide about 70 times. And then that aglet's gone. And then when that cell starts to divide, it's starting to eat into its genes. And when it starts to eat into its genes, your immune system says, hey, you're acting like a cancer cell. I'm going to kill you, boy. Or you start to kill yourself, called apoptosis. So each one of your cells basically has a fuse, a timer. It's called a telomere. It tells the cell when to die. Telomere protects the ends of your chromosomes. Every time the cell divides, the telomeres get a little shorter. If it gets too short, should be getting it while the getting's good. A little Too Short joke, you know, Too Short, rapper from the Bay Area from back in the day. If it gets too short, the cell will kill itself. Okay? So let's do a little example of this. So here we got a cell dividing. This looks so bad because it's so thick. I'm going to do this down at the bottom here. All right. This boy going to have shorter telomeres now. Okay. See how they're shorter? And then let's do one more round of cell division. And copy this bad boy and make this a little quicker. Not sure why that did that. Okay, and now we're going to have even shorter telomeres. Real short little guys. Boy's almost gone. Boop. This little baby boy, little baby telomere, just barely alive. And then we'll do any more division and the cells would die. These boys would be like ghosts because they wouldn't exist, because they wouldn't have telomeres. We'd start be eaten into their genetic material. Cell death. Okay, so your cells have a built-in, basically, time bomb saying when to die. And this actually protects you from cancer. It's also one of the reasons why we age. Okay? But our stem cells and cancer cells protect their telomeres. Okay? They express a protein, it's an enzyme, called telomerase. A's, remember that? It's an enzyme that extends their telomeres. Telomeres. Okay? So, it keeps their telomeres at length. And this is like happening with your stem cells, right? Down, like let's say your basal cells in your skin. You want to keep them dividing a lot longer than like this cell in between, right? So your stem cells maintain their telomere length by expressing telomerase. By expressing telomerase. Okay? So for a cell to become cancerous, oh, it's most certainly expressing telomerase. If it's going to survive a long time, otherwise, eventually, what would happen is these cells would run out of telomeres and then they would just die. So a cancer is most certainly expressing telomerase. Okay, this is also one of the reasons why we age. After a while of aging, even if you're expressing some telomerase, you have to express a bunch, your cells are gonna start to die. Unless you're expressing a ton of telomerase, which Henrietta Lacks cells are doing. Why? Her cancer cells express a lot of telomerase to keep her chromosomes protected. Okay? You guys now have a graduate level understanding of cancer. Most people have no idea what any of this stuff is. And this also has to do with your aging. If you look at a mouse that looks really old, and you look at it, it's old, and you look at its telomeres are shorter. Right? Doesn't this make you kind of want to be like, how do I turn on my telomerase in my cells? But be careful, because then you might get cancer. Like it's a double-edged sword, man. We're dancing with the devil with this stuff. Okay. Her cancer cells express a lot of telomerase to keep her chromosomes protected. Protected. All right, so they can live forever. Telomerase. Okay. All right. There's something else I wanted to get into with this. Yeah, this is one of the reasons why we age. Oh, yes, that's what I want to talk about. Okay, remember when I talked about this cloning up here? Right? This is cloning. We take the nucleus of an adult cell, in this case a tadpole, an adult cell, and put it into an egg, and then it'll grow into an adult, right? Because it contains all the instructions. But this sheep that we did this with only lived to be about 6. I don't know how long the average lifespan of a sheep is. Let me look that up real quick. Average lifespan of a sheep. Okay, 10 to 12 years. So she only lived about half of her lifespan, right? And why would that be? Well, because they took the nucleus of an adult cell, which is going to have some shortened telomeres. Likely. Like this is likely what's happening. Like they, telomeres like this or like this. So I'm sure that some of the telomerase in the egg is going to extend those, but it's not going to be able to extend them fully to where they used to be. And so that's one of the reasons potentially why Dolly only lived to be a certain age. Or not as long as she could have lived. Could be other things too, but it's incredible what is happening in our bodies. And I hope that you got something out of this class that changed the way you see the universe. That's my number 1 goal. Number 2 goal would be something that you can take with you in your everyday life. And that can be useful. it's been a pleasure teaching you guys. I mean, I haven't wanted to do this every single day that I've had to do it. It's a lot of work to do these lectures all at once. But I did really enjoy it. And I hope that you got something out of this. All right. Good luck on your next adventures. Let me know if you need anything. I'm here. I'm not going anywhere. Later.